Zika virus disease is caused by a virus transmitted primarily by Aedes mosquitoes. People with Zika virus disease can have symptoms including mild fever, skin rash, conjunctivitis, muscle and joint pain, malaise, or headache, which normally lasts for 2 to 7 days. However, 80% of the people do not experience any symptoms at all. Zika may lead to dangerous nerve conditions in adults and may be transmitted from a pregnant lady to her unborn fetus, which may lead to microcephaly and other deadly congenital abnormalities in her infant. Microcephaly is a poorly understood congenital condition in which babies are born with an unusually small head due to damaged brain tissue. Although exactly how this condition may affect the baby's life in the future is still undetermined, early case studies suggest that they could have serious brain damage which may be fatal. Unfortunately, there are still no cure or vaccines available. So what is Zika?
Virus is part of the genus of viruses called flavivirus, which also includes viruses such as dengue virus, the yellow fever virus, and the West Nile virus. The viral RNA genome, which is carried by the capsid, is enclosed by the pre-membrane, which in turn is surrounded by an icosahedral outer envelope.
Virus infection enters the body by the blood of an infected Aedes mosquitoes. It enters the bloodstream and is transported all around the body. It gains entry into host cells through the interaction of virus envelope glycoprotein with the host cell surface receptors. The four receptors are DC sign, TYR3, TIM1, and most important AXL receptor. A tyrosine kinase which mediates the inflammatory process, therefore playing a major role in the Zika transmission. Hence, in this video, we will focus on the AXL receptor. TAM ligand gas6 acts as a cofactor to recruit virus into the AXL receptor. This results in the internalization of the Zika virus through clathrin-mediated endocytosis. Zika virus containing vesicles translocate to RB5+ endosome and induce a transcription of toll-like receptor TLR3. TLR3 is part of a family of pattern recognition receptors of the human innate immune system where they are expressed on the surface and in endosomes of many cell types and are able to recognize microbial structures such as endotoxins and viral RNA. They are then able to transduce signals that lead to the expression of inflammatory and antiviral genes, defending against invading viruses. However, when the Zika virus RNA binds to a TLR3 receptor, it upregulates the transduction of signals made by TLR3. In other words, it causes TLR3 receptor to be oversensitive to stimuli. This results in the hyperactivated TLR3 receptor to turn off genes that stem cells need to specialize into brain cells and turn on genes that trigger cell suicide, furthermore disrupting mitosis. While it hijacks TLR3 receptors, it also transcribes other interferon-stimulated genes that play a role in sensing viral infection, DDX58 and IFIH. This capability leads to the Zika virus' abilities to cause neurogenesis, which is the reduced number of brain cells produced, resulting in incomplete growth of the brain and thus leading to microcephaly in infants, hence resulting in the suppression of our innate immune system and subsequently productive infection. In a productive infection, the pro-virus is transcribed into the messenger RNA, which directly produces new virus which in turn infect the other cells via the lytic cycle.
The placenta is composed of several layers of cells and has unique structural, cellular, and immunological properties which allows it to act as a barrier against bacterial and viral infections. However, Zika virus antigen and RNA were found to be present in the amniotic fluid and placental tissues of Zika infected women and their newborn's brain tissue, who has been diagnosed with microcephaly. This shows that the pathogen gains access to the fetal brain during pregnancy by crossing the placental barrier. The Zika virus passes through the placenta to reach the fetus by directly infecting placental cells. It mainly targets the Hofbauer cells and the trophoblasts. Hofbauer cells are fetal placental macrophages located adjacent to fetal capillaries, and they have been known to play a role in protecting the fetus from pathogens. On the other hand, trophoblasts are cells forming the outer layer of a blastocyst which provide nutrients to the embryo and develop into a large part of the placenta. The damaging and infecting of both the Hofbauer cells and the trophoblasts leads to induced apoptosis of trophoblasts and vascular damage, resulting in the disruption of the placental barrier, compromising its protection against viral infections, consequently conveying Zika virus to the fetus.
Placental insufficiency, which is resulted from vascular injury, reduced number of capillaries, irregular shaped capillaries, and apoptotic trophoblast leads to intrauterine growth restriction. In addition, inflammatory responses such as the increased levels of cytokines such as IP10, IL-6, IL-8, VEGF, MCP1, and GCSF when exposed to fetal brain causes cell death and dysregulation of the differentiation and proliferation of NPCs. These two factors, placental insufficiency and inflammatory responses, lead to microcephaly in unborn infants. These are the mutations in the Brazilian Zika virus strain that lead to higher chances of getting microcephaly. In present day, a new study suggests that most of the children born with brain abnormalities caused by the Zika virus are facing severe health and developmental challenges at 2 years of age. This shows that not only does more research have to be done to find a cure and vaccine for the prevention of the Zika virus, but also planning for the long-term care of these babies using these findings has to start now.